Hey guys, Miss Cote here, and we're continuing learning about protein synthesis. In the previous video, we looked at the first stage of protein synthesis, making a messenger RNA transcript from a DNA template. This video will finish off the process by taking the messenger RNA and translating it into a polypeptide. So let's get started. To review the central dogma of molecular biology, the correct order of the flow of genetic information is DNA to RNA to protein. Going from DNA to RNA is the process of transcription, which was the focus of the last video. At the end of the last video, we had processed and made a mature messenger RNA that's ready to leave the nucleus and find a ribosome. The second stage will be to go from messenger RNA to the protein, or technically a polypeptide. This is the process of translation. One of the big questions was, how do we know what the sequence of amino acids are in a polypeptide from a bunch of A's, U's, C's, and G's? How can 4 bases code for 20 amino acids? The answer is that the genetic code is based on a series of messenger RNA base triplets called codons. From the 4 different nucleotide base pairs, there are 64 different triplet combinations, shown in this chart. 61 of them code for amino acids, with one of them, shaded in green, designated as the start codon. Every polypeptide begins with this codon in the amino acid methionine, usually abbreviated as MET. The other 3 codons code for stop signals to mark the end of translation. There's no amino acid for stop. The sequence of amino acids just ends. For those of you who are not familiar with using a chart like this, I think most of you probably used the circle chart in your other biology class. You find the first letter from the left-hand side, the second letter is across the top, so find the appropriate box, and then the third letter is found from the right side of the chart. Please note that due to the fact that there are 20 amino acids and 61 codons, you will see some repetition or redundancy in the code. More than one codon may specify the same amino acid. Usually it doesn't matter what the third letter is. For example, CC whatever will code for proline, or AC anything will code for threonine. This can become important in the case of mutations. A mutation in a third letter of certain codons will not change the amino acid, and this is considered a silent mutation. I'll be working hard over vacation to add a mutations video to the collection. This genetic code is nearly universal, meaning that it doesn't matter if you're a bacteria, a carrot, or a mammal. This code works for pretty much all species of organisms. So scientists are able to insert genes from one species to another and be able to express the protein, since this genetic code works for all organisms. For example, the gene that causes a firefly's butt to glow can be transplanted into a tobacco plant and cause the tobacco plant to glow. Or a gene that makes jellyfish fluorescent was injected into fertilized pig eggs. This is one of the pigs, which expresses the fluorescence in parts of its body. Before I move on to the process of translation, I just want to put it out there to make sure you guys know how to use this chart. College Board will give you questions where you not only need to go from messenger RNA to amino acids, but they can also give you an amino acid sequence, and you may be asked to find a possible messenger RNA sequence that could code for that amino acid sequence. All right, time to move on. If you haven't figured it out by now, translation is using the instructions in the messenger RNA to create the polypeptide sequence. There are many players involved in translation, and so far we only have the messenger RNA. So let's look at the next important piece, the T or transfer RNAs. These are the RNAs that are responsible for bringing the amino acids to the ribosome. So over on the left is a molecule of transfer RNA. It is single-stranded, like RNA is, and it's made up of about 80 nucleotides. Because of the length of the 80 base pairs, there are areas where the base pairs are complementary and it folds into this upside-down 3-leaf clover shape. It also has the ability to twist into this 3-dimensional structure. So your book represents it as this blue shape over here on the right. I've seen it represented this way on College Board questions as well. At the top of each transfer RNA is the amino acid binding site. These molecules of transfer RNA are not identical. Each transfer RNA is specific to the amino acid it carries. So assume there are 20 different types of transfer RNA moving around the cell, one for every amino acid. At the bottom of the transfer RNA is a sequence of 3 nucleotides called the anticodon. The prefix anti means opposite or against, so these would be the opposite of the codon. The anticodon is complementary to the codon. The function of the anticodon is to bring the transfer RNA to the correct codon on the messenger RNA, with the correct amino acid that the codon codes for. While I would know what the anticodon is, I wouldn't stress too much about it. College Board focuses more on the codon in the amino acid it codes for. I find kids overthink the role of the anticodon. It messes them up and gets them even more confused. So how does the amino acid attach to the correct transfer RNA? Let's take a look. There are enzymes that form the covalent bond between the amino acid and the transfer RNA. Each amino acid transfer RNA pair has a specific enzyme, but we just generically refer to the enzyme as amino acyl tRNA synthetase. If you remember from learning about enzymes, the enzyme has an active site that is specific to the substrate being catalyzed. In this case, we're joining 2 molecules together, otherwise known as an anabolic reaction, which requires energy. As you can see, the shape of the active site puts the 2 pieces in the proper orientation so the bond can form quickly, thus speeding up the reaction. Now the transfer RNA can bring the amino acid to the ribosome. We keep mentioning the ribosome, but haven't really looked at it in detail yet. Back in Chapter 6, Parts of the Cell, you basically learned that the ribosome is the site of protein synthesis. Ribosomes are made up of proteins and R, or ribosomal RNA, and exist as 2 subunits, large and small. It's only during translation when the 2 pieces come together. And while both prokaryotes and eukaryotes have ribosomes, they are slightly different from each other. Different enough so that when you have a bacterial infection, antibiotics can target bacterial ribosomes and shut down those ribosomes, and therefore protein production, while not harming our ribosomes. So here's what a ribosome looks like. The large subunit sits on top of the small subunit. Notice that there are 3 binding sites on the large subunit. Let me explain what the function of these sites are in the next diagram. The P site, the one in the middle, holds the transfer RNA that is carrying the growing amino acid chain. The A site over on the right is where the new transfer RNA comes in to bind with the next amino acid. And the E site is the exit site, where the transfer RNA that gave up its amino acid chain can leave and go pick up another amino acid. Now let's put it all together. Just like in transcription, there are 3 stages to translation: initiation, elongation, and termination. So let's start with initiation. To start building a polypeptide chain, you need the messenger RNA, the transfer RNA with the first amino acid, methionine, and the small subunit of the ribosome. These pieces come together, and the ribosome slides down the messenger RNA until it reaches the start codon, which is AUG. Notice that the start codon is not necessarily at the beginning of the messenger RNA. Then the large subunit comes in and binds. This is the translation initiation complex. During elongation, the amino acids are added one at a time to the growing polypeptide chain. Let's go through a cycle. So here at the top, we have a transfer RNA with an amino acid chain in the P site. The next transfer RNA with its amino acid comes into the A site, where the anticodon on the transfer RNA matches with the codon on the messenger RNA. Once that the 2 transfer RNAs are in their sites, the peptide bond can form. The transfer RNA holding the chain in the P site will hand over the polypeptide chain to the new transfer RNA in the A site. The ribosome slides down the messenger RNA to the next codon. The transfer RNA that was in the P site is now empty and leaves to go off to find another amino acid to come back and bring that one in. The transfer RNA that's now holding the polypeptide chain slides into the P site, and the A site is empty. Now the cycle can start over again to add another amino acid to the chain. Notice that the codon recognition and translocation steps require energy in the form of GTP. At some point, the ribosome will come to a stop codon. Instead of a transfer RNA, another molecule called a release factor has the anticodon to match the stop codon. And instead of an amino acid, the release factor has a water molecule to add to the polypeptide chain. So when the transfer RNA in the P site gives up its chain to the release factor, the reaction releases the polypeptide, the rest of the translation complex comes apart, and translation is officially finished. Now that the amino acid sequence has been created, remember that does not make it a functional protein. That polypeptide, which is in the primary level of structure, needs to be folded into the secondary, tertiary, and possibly the quaternary level. Also, any modifications, such as adding a carbohydrate or lipid, is done at this time as well. Now the protein is in its functional form. Remember, there are two types of ribosomes, free and bound, and the difference between the two depends on the type of protein they are synthesizing. If the protein will be used in the cytosol, then it's made on the free ribosome. However, if the protein is destined for secretion, then it needs to be made on a bound ribosome so it can enter the endomembrane system. All polypeptides start on a free ribosome. So if that ribosome needs to become bound, how does it get there? Any polypeptides that need to enter the endomembrane system have a signal peptide at the beginning of it, shown in green. This signal peptide is recognized by a signal recognition particle, or SRP for short, and the SRP will bind to and tow the entire translation complex to a pore on the ER, where the synthesis of the polypeptide can continue and enter into the lumen of the ER. Eventually, it'll break off into a vesicle and travel to the Golgi for modification and eventually end up outside the cell or in the cell membrane. The last thing I want to touch on is that if the cell needs to make many polypeptides, it can do so by having multiple ribosomes work on the same messenger RNA at the same time. This complex is referred to as a polyribosome. And remember, in bacteria, since transcription and translation can happen simultaneously, having a polyribosome can really increase the amount of polypeptides being made, which makes sense. Since bacteria multiply way quicker than our cells, they probably need to produce proteins at a higher rate than we do. So I know this was long, no frills, and I probably spoke way too fast, but it's finally done. So until next time, be a good human.